Imagine you're sitting at your desk and somebody knocks on your door and they hand you a bucket that's filled with fresh human skin. Ugh. Welcome to our lab, where we receive donations of human tissues like skin that's left over after surgeries, and it would otherwise be thrown away. This may seem disgusting or gross, but it's actually amazing because human tissues, those are powerful tools for studying human viruses. Myself, I study dengue virus, HIV, and SARS-CoV-2, and I'm primarily interested in understanding how our immune systems see these viruses, so I can help defeat them. And in order to understand this, I'd like you to imagine a chess set, Queen's Gambit style, but I'll help. So when a virus plays your body in biological chess, it's playing white, making the first move by infecting you. But let's focus on the black army, your immune system. We often think of our immune systems in the singular, but they're actually composed of a lot of different moving pieces. For instance, we have the pawns. Those are the dendritic cells, which patrol the perimeters of your body, like in the top layers of your skin. And they may seem benign at first, but as soon as they notice an invader entering the field of play, they spring into action and alert their comrades that it's time to prepare for battle. Next, you'll see cells that, like bishops, roll out predictably, but quickly, to hopefully give our side, the black team, an early advantage. Those ones are called, and I kid you not, natural killer cells. Next come cells that, like the queen, can make efficient and far-reaching attacks. Those are the B cells, which produce antibodies that spread out over the entire field of play, and they pick off viruses, one by one. Now, this isn't a comprehensive overview of all of the immune cells or all the chess pieces, but you get the idea. Each of our immune cells is unique, and they all have a role to play in the battle of the host and the hijacker. And that brings us back to the skin. Because often in the lab, scientists will only look at how immune cells or viruses interact with immune cells one cell type at a time. And that gives us important information, but it doesn't give us the same insight as if we look at the whole board. And that's how what we do is different. By looking at the whole chess set, we can see how viruses react when they're faced with an immune army. And by using human tissues to do this, instead of lab rats, for example, we're using the most realistic possible system to assess this battle and see where we can help these guys out. Because our ultimate goal is to find new and more accessible ways to treat the viruses we study by giving our immune systems a little help. So those buckets of skin, they're not just a great conversation opener. They're also our way to say to the virus, check, mate.
Brilliant work. Thank you very much, Alex. Just a very quick dip in before the questions, just to make sure that everybody is still recording. If you could check and give me a thumbs up. Nicely done. So I'm going to kick off the questions with Julia.
Thanks so much, Alex. I wonder whether I can ask, how much do we know about how the natural killer cells talk to the B cells, the bishops to the pawns and so on, and the impacts of that?
Yeah, really interesting question. So to be honest, I actually don't focus on natural killer cells in my immune system. I chose them because of in my immune system. I hope that's using natural killer cells. In my research, I chose them because of the awesome name. But what I wanted to showcase is that there are different sort of sectors to the immune system. So natural killer cells, those are the first defenses, part of the innate immune system that kind of always responds. And all these different cells in the innate immune system, they basically send out biological letters called cytokines that will spread out throughout the body. And then they activate specific second responders like B cells and also T cells in a way that's suited to whatever the particular opponent that they have detected is. So actually it really depends on whether it's a virus, a bacteria, a creepy crawly parasite, and it's a huge research field. So we actually do know quite a lot, yeah.
Alex, thank you so much. That was fascinating. So one thing I loved about what you had to say was how you looked at the whole immune system. So you weren't looking cell by cell or at the different elements of our immune system. How is that does that require bringing together different disciplines within the work that you do, or is it just a different frame that you set the research in?
Yeah, I would say it's a bit of both. So I like to say that my research is at the intersection between infection and immunity. Oftentimes people will focus more on viruses or more on the immune system, and we really try to bridge the two and look at the connections. And in terms of setting the frame, yeah, I would say, I mean, the chessboard does have some limitations. I would say we're using as much of the chessboard as possible in an ethical and, I like to think, quite environmentally friendly way as well, kind of turning this trash tissue into treasure. So we're basically setting the frame as wide as possible and bridging all sorts of different fields to get the most realistic answers, to get the closest to the truth.
Thank you.
Thank you so much, Alex, for this wonderful talk. I have something in my mind that I'm curious for a long time. For example, when we are infected with COVID, how many particles do it's necessary to make us ill? I mean, if there is just one particle, most of us will be able to, I don't know, to kill it. Do you have an idea that this is known or not?
This is a super interesting question that people are still looking into for COVID. What's interesting about this is it's completely different depending on the virus or the bacterium. For instance, I'm quite sure with Mycobacterium tuberculosis, causes tuberculosis. If you get one bacterium, you can develop tuberculosis. With most respiratory viruses, like SARS-CoV-2, you usually need a bigger dose. Like if you walk through a cloud of particles from somebody's cough, you're more likely to get infected than if you're outside and there's one lonely virus drifting along that gets onto you. Usually your immune system will be able to clear that for viruses related to SARS-CoV-2 at least. But that's still an ongoing question for our latest SARS.
Thank you.